I'd like to thank Charles for giving me the opportunity to come here to Korea to visit for the first time. It's wonderful, and I also really appreciate being included in this important symposium today, so thank you. I'm going to talk about modeling Mendelian diseases in mice using both forward and reverse genetic approaches. My lab is part of a group at the Jackson Laboratory called the Genetic Resource Sciences Department, and so our mission is to create new mouse models of disease. And so we do this in a variety of ways, by creating new strains, by using new technologies to credential existing strains, to improve them, to standardize phenotyping, and all of these efforts feed into our JAX repository, which is a collection of now over 8,000 genetically engineered mouse models and associated resources like, for example, embryonic stem cell lines and adult stem cell lines. And so why do we care about using mouse models? And so most of you are aware of using mice for genome functionalization. So the idea here is to understand gene function through using mice. We can also use mouse models to understand basic biology. And finally, we can use mouse models for the early stages of drug development. And so what I'm showing you here is an example of a mouse model that was actually created by my predecessor at the Jackson Laboratory, Dr. Muriel Davison. She created this Down syndrome mouse model, which we call affectionately TS65. And this is actually one of the most widely used mouse models of Down syndrome. And it was used most recently for the early preclinical work on memantine in Down syndrome. So a couple of approaches. This is just some background to forward and reverse genetics. Some of you might already be familiar with these approaches. But here, forward genetic approaches is where you start with an animal that has a phenotype of interest. So this is an example of an obese mouse here. And the idea is to use genetic mapping and sequencing to understand the genes that are responsible for that phenotype. Reverse genetic approaches involve starting with the gene. And so in this case, you might have some idea of what a gene might do or that it might be involved in a particular disease. So you can use genetic engineering to modify that gene, and then you can study the resulting phenotype. So I'll start by talking a little bit about how we use forward genetic approaches. So forward genetics has the advantage of being unbiased, and so this is really the way that we can make novel gene discoveries. And so it relies on mutation rates, and mutation rates in mice, spontaneous mutation rates, occur in about 1 in 500,000 individuals per generation. You can increase that rate using mutagenesis, so irradiation or chemical mutagenesis, and that can increase the mutation rate to about 1 in 1,000 per locus per gamete. And so you can see that the disadvantage here is that you're relying on serendipity. You don't have any control over where those mutations occur. And so this can be a slow process unless you happen to have a huge vivarium of mice available to you. And as Dr. Lee explained at the Jackson Laboratory, on any one day, we have about 1 million mice live on the shelf. And so that gives us a huge advantage to use just spontaneously arising mutations as a way to do forward genetics. And a couple of the advances that really changed for us the way that we map genes. These are actually old chromosome maps, sort of the old-fashioned way that we would identify genes just by linkage. But when the mouse reference genome was published in 2001, and then later high-throughput sequencing was first published and started to be used, we had now a huge advantage of being able to quickly go from a mouse phenotype to a gene of interest. And this is just to give you one example of the kinds of phenotypes that we see. So here is a mouse model of, I'm going to skip ahead. That was my slide that I was supposed to delete. So this is an example of a mouse model that has a hyperkeratosis phenotype. This is a model of Pachyonychia congenita. And this is actually a very painful hyperkeratosis condition. It's a rare Mendelian disorder. And you can see in the mouse model, this hyperkeratin phenotype in the nails that is also seen in people with this disease. And this is just to give you an example of all the phenotypes that we get just using our forward genetics approaches. A large percentage of the phenotypes we have are visible phenotypes, and this is because we rely on the people who take care of the animals on a day-to-day basis to recognize these deviant phenotypes. It's part of our genetic control process to remove these phenotypic deviants from our breeding colonies. These animals are sent to our mouse clinic, and at the mouse clinic we determine if these phenotypes are heritable. About half of them are heritable, and when they are, then we become very interested in identifying the genes that are responsible. So what you see here, the largest percentage of the phenotypes that we see are behavioral and neurological, because as you can imagine, those are very easy to see. Then we have the integument phenotypes that affect skin and hair, craniofacial, skeletal phenotypes, etc. And then the smaller proportion of phenotypes you see up here are those that take a little bit more work to identify. So up here we have, for example, the reproductive phenotypes, vision, eye phenotypes, metabolic phenotypes. So when I started in our group, I really was interested in using exome sequencing to find these mutations. And at the time, we didn't have a way of doing mouse exome sequencing, so we worked on developing a mouse exome that would allow us to sequence around 200,000 coding regions in the mouse genome. That represents 33,000 genes and microRNAs. And this is just an example of what the data look like. So in gray, you see the high throughput sequencing data mapped to the reference genome. And here are exons across the bottom. And you can see we have very high coverage of the coding portions of the genome. And then I also want to show you here that we get coverage in the flanking intron sequence too. And that's important because you often see mutations in splice acceptor donor sites that can lead to phenotypes. And this is sort of a complicated slide, but this is just to let you know that in order to analyze the data, you have to have some computational ability. And the idea, what we do here is we just take the sequencing data, we map those data to the mouse reference genome, and then we call variants. And so the variants that we find are single nucleotide variants, or they're small insertions and deletions that are less than 50 base pairs. I think it's important to look at this too. So when we look at any individual exome, we see a large number of variants, but the number of variants that we see depends on the strain background that we're looking at. So as you can see at the top here, this is C57 Black 6J. This is the same inbred mouse strain that the mouse reference genome was sequenced from. So whenever we have a mutation that arises on this strain background, we get less than 1,000 variants, which is expected since these animals are closely related to the mouse reference. But if you look at these other strains and pay particular attention to these wild-derived substrains down here, we see over 300,000 variants every time we sequence one of these exomes. And you can compare that to a typical human exome where we see around 25,000 variants. So what we wanted to do to start our study was to take about 200 of these mutants and use exome sequencing to see how many of the mutations that we could find using exome sequencing. And when we did that, we had about 1.2 million variants from 200 exomes. And so here is just a look at what our variant data look like. If you look at the frequency of any individual variant across our entire data set, you can see some of the variants are very rare, and some of them are very common. So we first wondered, well, what are all of these common variants that we keep finding over and over again every time we sequence an exome? And so what we did was we took about 300 of those variants that we found in 100% of our samples, and we compared them to the mouse reference genome, and we found that more than half of them actually mapped to regions of the mouse genome where there are reported errors in the reference assembly. And so we think that the other half probably identify other errors in the reference assembly that haven't been identified yet. When we look at the variants that are more rare, we assume that these are probably the variants that are causing the phenotypes that we're seeing in the mice, because we see them in the best case scenario only in one sample in the individual mutant that we sequenced. So this is to give you an example of how we look at the data. This is looking at one individual mutant. This is a mutation that arose on BALB-C, which is a background that is a little bit further removed from the mouse reference genome. So we have in this individual exome about 134,000 variant calls. So how do we go from there to what we think is a single causative mutation? And so what we first do is we look at only those variants that are in regions that have sufficient coverage. We like to have at least 5 reads coverage before we are confident in our variant calls. And then we look at only those that are novel. So if we have a variant that is present in publicly available SNP databases, then we know that it's a variant that's been identified before by some other project and probably isn't causing our unique phenotype. So we only look at those that are not in the publicly available SNP databases. Then we take the subset of those that overlap with the map position. So we do genetically map these mutations. We map them to a chromosome. We don't map them any further than that, but just getting some chromosome linkage helps us narrow down the field quite a bit, as you can see here. Then we take the subset that has the allele ratio we expect. What this means is that the variant call is going to be either heterozygous or homozygous, and we know that the animal we sequenced was homozygous or heterozygous, so we make sure that we only look at those that make sense with the genotype of the animal. And then finally, we want to look at those that have a potential impact on the function of the protein. So is it a non-synonymous coding SNP? Is it a missense mutation? Is it in a splice site? And then from that subset, we look at those that are significantly rare, and that gets back to the slide that I showed you previously. We actually find that very few are present only in the single sample that we sequence, and so this allows us to get to that causative mutation fairly quickly. And in the case of this mutant, this MAP3K11 mutation actually was consistent with a previously published knockout phenotype of this particular mouse. So we can extend that analysis to our entire data set. This is getting back to the 1.2 million variants. We don't necessarily have to look at every individual exome. We can look at them as an entire group. And so we can do that all within a very short period of time. And in our entire group of 200 mutants that we looked at in our initial pass, we were able to identify causative mutations in 80 of them. So not all 200, but 80. And so we wanted to ask, well, how come we haven't found causative mutations for all 200 of these mouse models that we have looked at? And we went back and looked at the data, and we found another 11 mutations that were not single nucleotide mutations. They were not small indels. In fact, they were larger structural variations, akin to what Dr. Lee was talking about in his talk. And so in this case, we have in a few examples here, a 200 base pair deletion. We have a retroviral insertion. We have a large duplication, and here a larger deletion. But notice all of these are affecting coding sequence in some way. So after taking 200 of these, we identified in the end 91 pathogenic mutations, and I call them putative pathogenic mutations because you have to understand that in the mouse, the ultimate proof of a gene actually causing a phenotype, you'd have to go back in and recapitulate that mutation. So we still call them putative pathogenic mutations. These were occurring in 76 genes, and we found that about 10% of them were completely novel genes that have never had a mouse phenotype or a human disease associated with them before. We had several others that were mutations in genes that we knew something about, but the phenotypes that we had in our strains were different than what had been reported. And then over 80% of them turn out to be genes that are also associated with Mendelian disease in humans. And then, of course, for the example I showed you earlier, we found this mutation that causes hyperkeratosis phenotype. This turned out to be a novel keratin gene. So at this point, I want to give you a case study. This is one of the novel genes that we found. This gene is DDX10. We found a missense mutation in this gene that causes an autosomal recessive craniofacial phenotype. So you have this cute little mouse right here with a very short nose. Actually, it has a split nose. And we were interested in finding out whether or not there had been any reported cases of human diseases associated with DDX10. There was nothing in OMIM, but we did a literature search, and one paper we found reports a deletion on chromosome 11 causing craniofacial dysmorphology and learning deficits in this young girl. And this turns out to be a deletion in a large region that encompasses several genes, with DDX10 as one of those genes. In another paper that actually was published right around the same time that our paper came out, this is a patient that has a smaller deletion, 170 kb deletion, also in the same region of chromosome 11, and this actually only contains DDX10. And so this is a patient who has craniofacial dysmorphology, and he also has learning deficits as a young child. And so it's very interesting to see in the literature these couple of cases where we have examples of candidate genes from human studies that don't have enough data yet to support their candidacy, and we can compare those to some of the mouse phenotypes that we're finding. But one of the things I want to point out is we have no idea what the learning deficits are in these animals because we haven't done that phenotyping yet. But these data lead us to be able to do those studies now. The other thing I want to point out is that this is an autosomal recessive mutation. It's a missense mutation. The patient mutations are probably haploinsufficiencies because they're deletions. And so another thing that we'd like to do is go back and actually recreate the haploinsufficient mutation. And so this leads me to reverse genetic approaches. So at this point, now we have the ability to go back in, we have a candidate gene, we want to create a specific mutation in that gene. So just to give you an overview of reverse genetics, this is again starting with a gene and creating mutations in that gene to get to a phenotype of interest. The advantages obviously are that you're able to start with your gene. You can create an intelligent design, so I can say, I want to make this deletion. I want to create a haploinsufficiency, so I can go ahead and do that. It utilizes sophisticated genetic engineering, which we're now able to do. The disadvantages are that sometimes you can get unexpected phenotypes. So you might start with a gene that you think is really important, and then you create a knockout, and the phenotype is completely different than what you expect. So it does require some previous knowledge of that gene. And again, a couple of advances that have really changed this process are, again, the mouse reference genome, which was published in 2001. And then in 2012, the advent of targeted endonuclease technologies, CRISPR-Cas9. This is an example of a large-scale reverse genetic project, which you've heard about before. This is the Knockout Mouse Project, the International Mouse Phenotyping Consortium. And the idea here is to create a knockout of every single gene in the mouse genome, starting with ESLs. And so the comp centers that are around the world are creating these mice and doing first pass phenotyping on them. And the idea is that the more sophisticated, detailed phenotyping would be done by individual labs like your labs here. But what we would like to do is to use CRISPR-Cas9 editing to actually create very specific human disease mutations. So to go beyond just making a knockout, we want to be able to make specific mutations. So this is just for those of you who aren't aware, the CRISPR-Cas9 gene editing system relies on a guide RNA and then this Cas9 endonuclease. The guide RNA targets the Cas9 endonuclease to a specific site in the genome where a double-strand break is created. The double-strand break can get repaired by non-homologous end joining, and that leads to a deletion. And this typically gives you a knockout, depending on where the deletion is. Or it can be repaired by homologous recombination. Given a donor oligo, you can actually create a precise edit, a knock-in mutation using this technology. And so what that's done for us is that typically to do reverse genetics to make a knockout in an ESL previously took about 2 years. Now using CRISPR-Cas9, we have about 6 months. And the idea is that you take the guide RNAs and then an mRNA encoding the Cas9, you inject those into a mouse embryo, and you generate your heterozygous mice from that process. The entire thing takes, as I mentioned, 6 months. And so what that's meant for us in the past year, as we've been approached by more and more human geneticists who have interest in making specific Mendelian mutations in mice, we've been able to use CRISPR-Cas9. And this is just to give you an example of the sort of bolus of mutations now that are coming to us to make in mice. The majority of these are for congenital heart disease, but we also have a few that we've made in collaboration with the Mendelian disease sequencing centers. So just to conclude, in the 6 years since whole exome and whole genome sequencing has been used for gene discovery in humans, over 100 causative genes for Mendelian diseases have been discovered. And similarly, our rate of causative gene discovery in mouse models has increased by about 10 times. Now we can also use in parallel gene editing technologies to create mutations in mice much more quickly than we were ever able to do before. And that's allowing us now to use a combination of both forward and reverse genetic approaches to create new mouse models. So I'd just like to conclude by thanking my colleagues in our department of GRS in the JAX repository, JAX Computational Sciences, and then our collaborators at the Centers for Mendelian Genomics, and of course our funding agencies, and I always have to thank our mice.